The acute inflammatory response begins immediately after tissue injury. In this animation, we will learn how phagocytosis and oxygen-dependent killing mechanisms participate in the acute inflammatory response. We will also discuss conditions related to defects within this process. Let's take a look at an injury. In response to an injury, three main processes take place. 1. Arteriolar dilation, which increases blood flow to the affected region. 2. Increased capillary permeability. And 3. Migration of leukocytes. Let's start by focusing on a laceration. Within the affected region, notice the bacteria, macrophages, and mast cells within the tissue and neutrophils circulating through the blood vessel. Let's take a closer look. One of the body's first responses to bacterial invasion is complement activation and release of C5a protein from the infected region. At the same time, tissue macrophages are one of the first phagocytic cells to encounter bacteria. The macrophages will begin to phagocytose bacteria. And release chemokines such as interleukin-1, 6, 8, and tumor necrosis factor, or TNF. Released complement C5a protein binds to its receptor on mast cells, inducing the release of histamine, prostaglandin, and leukotriene in a process known as degranulation. Histamine causes vasodilation and stimulates the redistribution of P-selectin from its normal intracellular stores inside endothelial cells to the cell surface, whereas cytokines secreted by tissue macrophages and other cells regulate the expression of adhesion molecules such as E-selectin. E and P-selectin ligands are expressed on the surface of leukocytes, including neutrophils. The interaction between E and P-selectin and their ligands allows for leukocytes to bind to endothelial cells. E and P-selectin ligand binding is a low-affinity interaction that can be easily disrupted by flowing blood. As a result, leukocytes will bind to the endothelial surface, detach as they are pushed forward by blood, and then bind again, resulting in leukocytes rolling along the endothelial surface. These weak rolling interactions slow down the leukocytes and allow them to later bind more firmly to the endothelium with the help of integrins. Leukocytes normally express integrins in a low affinity state. Meanwhile, TNF and interleukin-1 will induce endothelial expression of ligands for integrins such as ICAM-1. Chemokines from the infected region, such as interleukin-8, bind to proteoglycans of endothelial cells. When rolling leukocytes attach to interleukin-8, their integrins convert to a high affinity state. This results in firm integrin-mediated binding of the leukocytes to the endothelium, facilitating migration through the endothelium in a process called transmigration or diapedesis. After leaving the circulation, leukocytes migrate toward the site of injury by chemotaxis. The most common chemoattractants are interleukin-8, C5a, and leukotriene B4. Once at the site of injury, these leukocytes will phagocytose bacteria. Phagocytosis involves three sequential steps. In the first step, recognition and attachment, leukocytes express several receptors that recognize pathogens. The most important of them are toll-like receptors, or TLR, and opsonins, such as receptors for IgG and complement C3b receptors. The second step involves creation of a phagolysosome. The bacterium binds to phagocyte receptors, and extensions of the cytoplasm called pseudopods can form around it. The plasma membrane then pinches off to form a phagosome, thus enclosing the bacterium. The phagosome fuses with a lysosome granule, producing a phagolysosome. The final step is oxygen-dependent killing. Notice the particular enzymes and structures involved with killing bacteria once phagocytosed. NADPH is oxidized in the cytoplasm. Hydrogen ions from this reaction then enter the phagolysosome through proton pumps. NADPH oxidase transfers electrons from reduced NADPH to oxygen molecules, forming superoxide anions. Next, superoxide dismutase converts hydrogen ions and superoxide anions into hydrogen peroxide. Hydrogen peroxide then breaks down into hydroxyl radicals, which kill the pathogen. Neutrophils contain the enzyme myeloperoxidase, which in the presence of chloride converts hydrogen peroxide to hypochlorite, labeled here as OCl, a potent antimicrobial agent that destroys microbes. Now that we have discussed how bacteria are killed, pathological conditions that hinder this process will be reviewed. Chronic granulomatous disease is an X-linked disorder involving recurrent chronic infections of catalase-positive bacteria. Chronic granulomatous disease is caused by a defect in NADPH oxidase, which is responsible for producing superoxide anion. Without properly functioning NADPH oxidase, electrons from reduced NADPH cannot be transformed to oxygen molecules. Thus, there are no oxygen-dependent intracellular metabolites such as superoxide anions, hydrogen peroxide, or hydroxyl radicals available to kill catalase-positive bacteria. These bacteria, such as Staph aureus, contain catalase enzymes that destroy hydrogen peroxide normally produced by bacteria. On the other hand, catalase-negative organisms lack catalase enzyme and, as a result, do not metabolize hydrogen peroxide. Thus, hydrogen peroxide produced by the organisms themselves is converted into hypochlorite by myeloperoxidase and chloride. Hypochlorite is then able to destroy these catalase-negative organisms. This is why patients with chronic granulomatous disease are susceptible to recurrent catalase-positive infections. Myeloperoxidase deficiency is another inherited disorder. It is usually without clinical symptoms, but can result in chronic infections due to fungi. Myeloperoxidase deficient neutrophils retain their ability to kill most microbes because NADPH oxidase functions properly. This means that neutrophils can transfer electrons from reduced NADPH to oxygen molecules by NADPH oxidase and form superoxide anions, which are then converted by superoxide dismutase into hydrogen peroxide. Hydrogen peroxide is then converted to hydroxyl radicals capable of killing microbes. However, the capacity to kill certain fungi such as Candida albicans can be completely absent in myeloperoxidase deficient neutrophils. Leukocyte adhesion deficiency is a rare autosomal recessive disorder of several types, with type 1 being the most common. It's due to a deficiency of beta-2 integrins, which leads to recurrent bacterial infections and a characteristic delay in umbilical cord sloughing. LAD1 is characterized by the absence of low-affinity beta-2 integrins, CD11 or CD18, on leukocytes. This leads to the inability of leukocytes to attach to the endothelium, preventing migration from the bloodstream towards sites of inflammation.